Morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
Welcome to
warahmatullahi.
webinar on Applied Microbiology Series 43, the topic production of microbial enzymes through fermentation, held by Research Center for Applied Microbiology, National Research and Innovation Agency, BRIN, today, Wednesday, November 13, 2024. In this occasion, we are honored to welcome Dr. Ahmad Fathoni, Head of Research Center for Applied Microbiology, BRIN, our speaker, Dr. Muhammad Irfan, visiting researcher of Research Center for Applied Microbiology, BRIN, and also Associate Professor at University of Sargodha. And our moderator, Dr. Titin Haryati, researcher of Research Center for Applied Microbiology, BRIN. Ladies and gentlemen, next agenda is singing the national anthem, Indonesia Raya. Okay, ladies and gentlemen, next is opening remark from Head of Research Center for Applied Microbiology, BRIN. To Dr. Ahmad Fathoni, time is yours.
Oke, terima kasih Mbak Oci ya. Thank you everyone. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Honorable Speaker Dr. Muhammad Irfan of Associate Professor from University of Sargodha, Pakistan, and he's also Director of Department of Biotechnology at the university. Also, Bapak Ibu sekalian, everyone, thank you very much for joining us this morning. Sorry for the 15-minute delay because I had a morning meeting just finished. Thank you for waiting and also attending this webinar. So this time we are going to discuss and talk about microbial production of enzymes through fermentation. So if you can see here the key, I think the key technology is fermentation. Yeah, Bapak Ibu sekalian. So the key enzyme is one of the fermentation product, but I think the key technologies, the main key technology here we are going to learn, we are going to discuss is about fermentation. And as we know that in Indonesia, yeah, not only in BRIN, in Indonesia, we need this kind of expert in the fermentation field. Not many researchers in Indonesia who are expert in this kind of fermentation because maybe especially at the university lack of facility but in BRIN, in Cibinong and in Serpong we have fermentation laboratory. I think most complete facility that we can utilize, we can access together from university, even from industry. And here we invited Dr. Irfan. He who is expert in the fermentation technology from Pakistan, and we are going to learn about what he has been doing for his work at the university. So I hope that we can create more researcher in this field, in the fermentation field, because it can be crucial point to deliver our result from laboratory to industry. So there is a pilot scale. So lab scale, pilot scale, and upscale in the industrial scale. Until now, we have still the problem in the piloting scale. Yeah. Besides, most of us not familiar with the fermentation or with the fermentor. Also, few research or activity in the upskilling or piloting process. In now at the Research Center for Applied Microbiology, we have started. Yeah, we have started. Maybe in the previous institution also have done this, but we want to strengthen this kind of activity, and then next year, even next year, we want to have more research, more activity on how we scale up, how we pilot our result from lab to pilot before we introduce to the industry. Yeah, and this is crucial, fermentation technology. I think all biotechnology product generated or produced from the fermentation process. Enzymes, microbes, and other metabolites. Now, in the world, fermentation is the key. Yeah. I would like to say that one example, for example, now in Estonia or in USA, they produce milk, cow milk, yeah, from the fermentation, through fermentation. So they don't grow cow, they don't grow animal, they don't need to provide lots of land to grow the animal, cow, to produce milk. Now, they only build a building consisting inside the fermenter, and then they produce milk from the fermentation. You cannot imagine now, yeah. We have in other country they have done that, and in Indonesia, why not? Yeah. In terms of infrastructure, we have research grant. BRIN provides, and the one thing is that we need to improve or to increase the human resources, our researcher. So increase our capacity research, and then also we need to create young talented researcher in this kind of field. Yeah. So Bapak Ibu, I hope that we in maybe start next year, we can have more and more researcher or activity doing this kind of fermentation, whatever we want to use for enzyme production, for microbe or cell production, or metabolite production, whatever. We must have this skill in the fermentation research. Yeah. So today I hope that Dr. Muhammad Irfan can share his expertise, his work so far, that has been doing so far to us. And then please, Bapak Ibu, ladies and gentlemen, we can discuss anything with Dr. Irfan, because I think he's now in Cibinong, based in Cibinong. So please just visit to his laboratory. So his host is Dr. Trisanti, and also with the Dr. Ika in Serpong. So anytime, please contact him and then discuss, and hopefully we can set up collaboration in the near future, next year. Yeah. I think that's all, Bapak Ibu semuanya. Thank you. I hope you can gain benefit from this webinar this morning. And again, thank you very much, Dr. Irfan, for visiting us here in Cibinong, and then for everyone for joining us. And yeah, thank you. And I open this webinar officially. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Fathoni. Ladies and gentlemen, before we start the agenda, we will have a photo session first. To all participants, please activate your camera, and for the committee to take the screenshot. Okay, I will start count from first slide. Ready? 1, 2, 3. Next slide. 1, 2, 3. Third slide. 1, 2, 3. Okay, thank you. And I would like to remind to all participants, please fill the attendance form from the link provided by the committee in the chat box. Now we are going to start presentation and discussion session, which will be led by Dr. Titin Haryati. So Dr. Titin, time is yours.
Okay, thank you, Mbak Uci. Okay, ladies and gentlemen, good morning. In this session, we are going to webinar with title of Production of Microbial Enzyme through Fermentation to Honorable Head of the Research Center for Applied Microbiology, Dr. Ahmad Fathoni, to Honorable Visiting Research of Research Group Enzyme for Bioenergy, Dr. Muhammad Irfan. Okay, before we begin this webinar session, I will read the CV of the Dr. Muhammad Irfan. Okay, Dr. Muhammad Irfan, please. Okay, Dr. Muhammad Irfan is from Associate Professor of University of Sargodha of Pakistan. He have H index at 30, very high, and he has published over 235 paper published and 22 chapter book and 2 book published and many more. Okay, and okay, for time, Dr. Muhammad Irfan, I will give your time to presentation at about 45 minutes, and then maybe 20 minutes more for discussion. Okay, Dr. Muhammad Irfan, the time is yours.
Thank you very much. First of all, I would like to thank the administration of BRIN and the government of Indonesia for providing me opportunity to be here in Indonesia. And I would like thanks to Dr. Ahmad Fathoni, who is the director of Microbiologist Research Center in Cibinong. So thank you very much for hosting me as a visiting researcher and your group. And I would like to thank also to the group members like Dr. Trisanti, Dr. Ika, Dr. Ahmad Sintowi, Dr. Titin, Camelia Prida. They are all group fellows who are working in enzyme and fermentation technology at Cibinong. So once again, I thank to all of you. I'm sure I would have a good opportunity and enjoying my life and some time in Cibinong, Indonesia. It's a good opportunity to explore Indonesia, and I hope we do have some good relationship between Pakistan and Indonesia. Once again, thanks for your time, for giving opportunity. So, and good morning, everybody. I would like to start my presentation by sharing my screen now. Can you see my screen?
Yes.
It's okay for everyone?
Yes. Good.
This morning, as said by Dr. Ahmad Fathoni, fermentation right now is a key technology for the production of valuable compounds through valorization of agricultural material, agricultural waste material, and for utilization of microbes to produce valuable compound. So today's today's webinar is about microbial enzyme production through fermentation. So going into more detail, first of all, I would like to introduce my country, my city. Here you can see I am from Sargodha. Sargodha is located between our capital, Islamabad, and Lahore, is in between. So this city is also known as California of Pakistan. Why we call it California, Pakistan? Because the major product from this city is citrus. In Pakistan, almost 80% of the citrus is being produced from Sargodha. So that's why we are calling it California of Pakistan. And this city is also known as City of Eagles. Why we call this City of Eagles? Because in my city, we do have air base. You can see that in picture, the jet plane, airplanes, fighter jets. If you have opportunity to visit Sargodha, you can oftenly see these fighter jets roaming around in Sargodha. So that's why we are calling it City of Eagles. And here you can see just like malls, mall same like as Chibinong Mall, Chijiam, and some other like malls. So this city also has some interaction, same as like Chibinong, or similar to like Jakarta also. So as you all know that enzymes, what are the enzymes? Do you know about enzyme? What is it? Enzyme is biological catalyst. And if we call the one-liner about enzyme, we can call without enzyme, there would be no life. So why we are calling this no life without enzyme? Because enzymes are the worker bees that make things happen. If we look around, if we are looking ourselves, like plants, animals, human beings, everything, most in living things, all metabolic activities, they are controlled by enzyme. So that's why we are calling no life without enzyme. So everybody know about the enzyme, about fermentation technology, and in a enzyme, they are broadly categorized into 6 main classes. So hydrolases, oxygen reductases, ligases, lyases, and so on. So majorly all enzymes, they are categorized into different classes. Main classes are 6 main classes. So when we talk about further how to get these enzymes, what are the main sources of enzyme? Why we are working on enzyme? Why we need to do this? Because if you look at the global market enzyme value, the enzyme, they are categorized by here application and by type also. If you look at the enzyme market by application, you can see that here data is represented in 2 years, 2017 and 2024, right now, this years. So you can see that the color in light green indicate the color of data of 2017, and the color about blue indicate the data of 2024. And you can see that enzyme, they are very important for food and beverages, household care, bioenergy, pharmaceutical and biotechnology, feed, and other applications. So day by day, the demand of these enzymes is becoming more and more to make our things happening, to smooth line our lives. So that's why we are working on enzymes. So that's why enzymes are the key pinpoint issue in biotechnology. So we are trying to produce enzyme using different sources, using different technologies at large scale, from lab scale to large scale to meet industrial demands. So if we look at the enzyme by type, so what are the main key enzymes which are required by the world in large quantity? So in by type, proteases, carbohydrates, these are the most important enzymes which are being produced by fermentation technology and which have a global market in the world. So that's why we are more focused, we are more interested to produce these enzymes, because every industry is using enzyme. So right now in this era of digitalization and sustainable development, we are going to produce the enzymes through sustainable development goals to make our climate less effective. So if we are doing the same work with some sort of catalyst, So the catalysts, they are not environment friendly. So that's why we are moving towards enzyme, because enzyme, they are eco-friendly, they are environment friendly, they are biodegradable, they do not harm our environment. So that's why we are moving towards enzymes. So what are the main sources of enzyme? How we can get enzyme from different sources? In general, enzyme, there are three major sources. The first one is from plants, second one is animals, the third one is microorganism. If we look at the plants, we are not using plants. Almost 4% of the enzyme that could be taken from the plant sources. But if we are going to extract these enzymes from plant, when we are using plant as a source of enzyme, so these plants, they have competition with our food. So that's why we are not going to produce, to extract enzyme from plant sources. The same case with animals. If we are producing enzyme from animals, you can see that the animals, they have a good relationship with our food. We can take milk, we can take food, we can take meat, everything from animals. And these animals, they are very also helpful to maintain the ecosystem. So if we are going to extract the enzyme from animal sources, at certain times, these animals will be eliminated, and we do have some problems with our food shortages. So that's why we are not going to extract the enzyme from animal. The alternative of both plants and animal is the microbes. So why we are more focused on microbes as compared to plants and animals? Because the microbes, they have no competition with our food as compared to plants and animals. The second thing is why we are more focused on microbial enzyme, because microbes, they don't need any space for their growth. These microbes could be produced, could be growth in small bottles in a controlled environment. And that's why we are more focused to extract enzyme through microbials. And you can see that about 80% of the enzyme, they could be produced from microorganism. And further, if we are going towards microorganism, we can further categorize, like in microorganism, we have so many kind of groups like algae, like fungi, bacteria, viruses, microalgae, and yeast also. So then we have to select what is our major source, what kind of microbes would be suitable for us. And as far as our fermentation technology is concerned, so in this, we are using some sort of the microbes which are obtained from the grass lister. If you know about grass list, what is meant by grass? Grass means generally regarded as a safe microbe. So for fermentation technology, for the production of enzyme at lab scale, at industrial scale, we must use microorganism that is not pathogenic, that is safe for human consumption, that is safe for lab handling. So that's why we are using microorganism and using some other grass microbe for the production of an enzyme. So this is all about the sources of enzyme, where we can get these enzyme, from which sources we can get these enzyme. So moving towards next, what is the fermentation? How you can define fermentation? Because fermentation, in life sciences, fermentation have some other definitions. If you can ask from biochemists, can you define fermentation? The biochemist can define fermentation refers to as energy generating process where organic compound act as both electron donor and acceptor. So this term, this definition came from biochemist. And right now we are all microbiologists, and you can see if we are talking about microbiology have so many kind of fields like applied microbiology, medical microbiology, water microbiology, agriculture microbiology, soil microbiology. So they have wide variation in microbiology. So right now we are this webinar is about applied microbiology. This center is Research Center of Applied Microbiology. As an applied microbiologist, we can define fermentation as the growth of microbes in large quantities under aerobic or anaerobic condition within a vessel referred to as a fermenter or bioreactor. That is called fermentation. So fermentation have two different definitions with respect to their area of specialization. If you are a biochemist, you can define fermentation as energy generating process. And if you are a microbiologist, you can define fermentation as a cultivation of cells in large numbers in large vessels, large reactor. That is called fermentation. So this whole definition, both definitions, they are taken from Industrial Microbiology book that is written by Michael J. Waits, and this book is published by Willie Sands. So this is the reference of this book. So you can read more about fermentation. So this is a little bit about fermentation, how we can define fermentation. So what are the main types of fermentation? How you can classify fermentation? So the fermentation have different classification based on different parameters. As we all know that all people who are dealing with the microorganism, who are dealing with fermentation, they can know that when you are going to cultivate a microorganism, you have to do all kind of steps. The first step, if you are going to do microorganism cultivation, the first one is the mode of cultivation. How long you will cultivate your microorganism. So the same case is with fermentation. If you are going to cultivate your microorganism on the basis of cultivation, fermentation is categorized into three main modes. The first one is batch culture, batch fermentation. The second one is continuous culture or continuous fermentation. And third one is fed batch culture, fed batch fermentation. So in batch fermentation, that is called a limited time fermentation. If you are working on batch fermentation, that means you have to define the time period of fermentation. For a whole long time, you can do this fermentation. Like you are working fermentation for 24 hours, 48 hours, or 96 hours. When you fix the time, that is called batch fermentation. And if you have no time limitation, you are going to cultivate your microorganism for longer period, for 1 month, 2 months, 3 months, 4 months. That is a continuous time. This is called continuous culture. And as we all know that when a microorganism is being cultivated, every microorganism, they need their food for their growth. And for some time, the food is being, with the passage of time, the food is being eliminated from the culture, and you have to provide a new feed with the passage of time. And in that time, that is called fed batch culture. So this is the first classification based on the time period, based on mode of cultivation. If we categorize further on water activity, based on water activity, the fermentation is mostly categorized into two main groups. The first one is submerged fermentation. The second one is solid state fermentation. Later on, we can further categorize what is meant by submerged fermentation and what is meant by solid state fermentation. In almost all of the fermentation, we are performing two different fermentation. Either you are dealing with submerged fermentation, or you are dealing with solid state fermentation. The third is immobilized system. Immobilized system in which you have to entrap, you have to grab your cells by different immobilization techniques like cross-linking, like entrapment, like making some gels, making some covalent bonding to make some other material. So on the base of water activity, the major categories are two: submerged and solid state. The third categorization of fermentation is based on oxygen requirement. As we all know that all over the world, most of the living organisms, some they need oxygen for their growth, they are known as aerobic, and some microorganisms they don't need oxygen for their growth, so we can call it anaerobic. So if your fermentation mode is aerobic, you are supplying oxygen, that is called aerobic fermentation. And if you are not providing oxygen to the fermentation system, that is called anaerobic fermentation. So on the base of oxygen requirement, fermentation are categorized into two main groups, aerobic and anaerobic. The third mode of categorization of fermentation is based on nutrient metabolism, how nutrition they are metabolized. If we are talking about fermentation based on nutritional metabolism, so we on the base of nutritional metabolism, we can categorize into different stages like conserved growth, synchronous growth, dioxic growth, cryptic growth, solvatinogenic or acidogenic growth, homo or heterofermentation. So all these based on the metabolism, how microbes they utilize the carbon source, the nutrition available in the medium. So because in microbiology, we have defined fermentation as a growth of cells in large numbers in fermenters in reactor. So to provide growth, we must need a supplement to supplement nutrition. If we are not providing supplement nutrition, that means our growth is our microbe is not growing very well. So at industrial scale or at large levels, the growth media play a key role to produce their metabolites. The last one is based on the number of inoculums. We can further categorize fermentation on the basis of three groups, like monofermentation, dual fermentation, or mixed fermentation. In my previous slides, I have mentioned about enzyme production from microbes. So when we are calling microbes, that doesn't mean it's a bacteria. When we are calling microbes, that means it is bacteria, it is a fungi, it's a virus, it's a microalgae, it's a yeast. So many kinds of groups could be utilized as a source of a microorganism. So when we are providing inoculum as a single microorganism, that is called monofermentation. Let's suppose if you are trying to produce an enzyme from fermentation and your inoculum is bacillus, your inoculum is bacteria, that means it is a monofermentation. And if you are using bacillus, bacteria as well as fungi, that is called dual fermentation. And if you are using inoculum of bacteria, fungi, and yeast, that is called mixed fermentation. So this is all about the classification of fermentation based on different parameters. Another classification of fermentation is based on sterilization. What kind of media, sterilization method you are using? If you are using fermentation media for sterilization purpose, and then you can do fermentation, that is called aseptic fermentation. And if you are not sterilizing your media and you are just doing without sterilization, that is called non-aseptic fermentation. So based on every parameters, you can classify fermentation into different groups. In the last fermentation, we can also categorize based on their products. So what kind of products you are going to produce during fermentation? If you are going to produce the alcohol during your fermentation, then it could be called as alcoholic fermentation. If your main target is citric acid, then you can call it citric acid fermentation. If you are going to produce lactic acid during your fermentation, then you can call it lactic acid fermentation. So this means that we can classify, we can categorize fermentation based on their products, what kind of products you are going to produce. So this is all about fermentation, how you can classify the fermentation process. So moving further, in lab scale or at industrial scale, we are using two different kind of fermentation. Either we are using solid state fermentation or either we are using submerged fermentation. If we are more focusing about solid state fermentation, so each fermentation have its own advantages and disadvantages. There are some pros and cons of each fermentation. If we are dealing with solid state fermentation, solid state fermentation do have some advantages. So what are the main advantages of solid state fermentation? The first advantage of solid state fermentation is that the medium is simple, easily available, and inexpensive. So please remember in your mind, when we are dealing with fermentation, when we are dealing with fermentation at large scale, the cost is a key factor. We have to utilize all kind of material that is very low cost. So in case of solid state fermentation, our media, our carbon sources, they are easily available and they are inexpensive. This is the main advantage of solid state fermentation. The second one, substrate need less pretreatment compared to liquid media. The third advantage is contaminations are restricted since the moisture content is low. And the fourth one is forced aeration is often easier, and they have simplified and minimized downstream processing and waste disposal. And this is simple fermentation equipment. And by solid state fermentation, you can get high volumetric productivity. So all these are different advantages. These are the pros of these solid state fermentation. In addition, if every technology have its advantages, merits, and demerits, if we are look towards demerits or disadvantage of solid state fermentation, so the first disadvantage of solid state fermentation is that in this there's low moisture level that may restrict the growth of microorganism. So that means the solid state fermentation, the growth rate is little slow as compared to liquid fermentation. The second one is removal of metabolic heat is a problem in large-scale solid state fermentation. You cannot maintain heat level during solid state fermentation. And the third disadvantage of solid state fermentation is that this is very difficult in monitoring the process parameters. So process parameters, these are the key parameters to control fermentation. So what are the process parameters during fermentation? The main key process parameters, they are to control pH, to control temperature, to control aeration, to control agitation, to control oxygen levels. So all these are the key factors which can restrict your fermentation process. So this is all about solid state fermentation. When we are moving towards solid fermentation, so semi-fermentation also have its own merits and demerits. What are the major merits and demerits of semi-fermentation? We can also call it liquid fermentation. So first we will discuss the advantages of semi-fermentation. So in case of semi-fermentation, the process parameters, they are very easy to measure, because in science, everything you have to measure it. How much quantity, how much quality, how much level, how much concentration you are going to give your microorganism. So in case of semi-fermentation, all these process parameters can be measured and can easily be controlled. They have ability to control. And in case of semi-fermentation, there is even distribution of nutrition to the microorganism. You can easily distribute the whole nutrition to the microorganism, and you can easily provide oxygen to the nutrition or to the microorganism. And also the availability of high water content for the growth of microorganism. As you all know that the life require water. Water is much needed for the growth of microorganism. Every living organism, they needed water for their life. So if the water level is very low, that means the microorganism could not be grown. So in case of summer fermentation, availability of water is very high. So why we are providing high water content in semi-fermentation? Because fermentation, in case of semi-fermentation, water act as a media, water act as a place for metabolic activities. And through this water, we can control pH. Through this water, we can control temperature of whole fermentation system. So these are the advantages of semi-fermentation. But we do have some disadvantages of semi-fermentation. When we are talking about demerits or disadvantages of semi-fermentation, so the first disadvantage of semi-fermentation is that we can use expensive media and expensive equipment in case of semi-fermentation. And the second one is It says complex and expensive downstreaming process and difficulty in the waste disposal. Why? Because in this case, our water content is very high as compared to solid state fermentation. So if your water level is very high, you need high power consumption to rotate, to mix your nutrition, to distribute all oxygen and nutrition and temperature to control all these. You need high power consumption. And mostly for semi fermentation, we are using different kind of bioreactors like stir tank reactors. We can use some airlift fermenter. We can use some other fermentation system also for this process. So these both submerged fermentation and solid state fermentation, these are two major fermentation systems which are frequently used in almost every kind of laboratory around the globe. So that's why I'm here talking about submerged and solid state. If we can shortly compare the submerged and solid state. So these are different parameters. In case of, if we talk about by definition, in solid state, the major part is solid state. And in this case, microbial growth is on solid surface of the substrate. But in case of semi-fermentation, the microbial growth is in suspended mode. And in case of solid state fermentation, the growth of microbe is based on supported media. If we talk about the mode of cultivation, is in solid substrate, and in case of semi-fermentation, it is in a liquid media. And if we talk about microbial cells, the growth is adhered to the solid surface. But in case of semi-fermentation, the growth of microorganism is evenly distributed in the media. In case of solid state fermentation, the culture medium is not flowing, but in case of submerged fermentation, the media is always free flowing. But if we talk about the water content of solid state fermentation, it all depends on the type of substrate, which kind of substrate you are using. And in case of solid state fermentation, the water content may be 40% to 80% in certain cases. But in case of semi-fermentation, the water content is about 95%. And if we talk about nutrient distribution, they are not evenly distributed in the media. But in case of semi-fermentation, whole nutrition is evenly distributed. And when we talk about inoculum ratio in both techniques, in case of solid state fermentation, you need high inoculum ratio. But in case of submerged, you can use low inoculum level. And if we talk about agitation, in case of solid state fermentation, it is optional, may or may not be involved. But in case of submerged fermentation, agitation is essential. Why? In case of submerged fermentation, agitation is essential because it provides equal distribution of nutrition to all microorganisms, as well as this agitation can help to distribute heat throughout the media. And in this way, we can easily control the temperature, oxygen, as well as equal distribution of nutrition. That's why we are providing agitation to our submerged fermentation system. So this is all about comparison between submerged fermentation and solid state fermentation, which we are oftenly working in the labs. What are the main process of fermentation? What kind of process we can do? How to start a fermentation process? If you are going to develop a fermentation process at large scale, at lab scale, at pilot scale, you have to focus on these steps. The first step is your choice of microorganism. What kind of culture you are using? So you have to take the first step is stock culture. I will talk about later about stock culture, how to get this microorganism, where to get this. The first one is you can get stock culture, then you can propagate this stock culture to a shake flask, and after making a viable cells in shake flask, then you can move further to a seed fermenter, you can make inoculum. In this, in side-by process, you first have to make a media formulation, media optimization, what kind of carbon source is required for your microbial growth, what kind of nitrogen source is required, what kind of other minerals they are required. So to deciding your media formulation, this media formulation is totally based on two things. First one, it is totally based on the type of product, what kind of product you are going to produce. The second is what kind of microbes you are going to use in your fermentation. So the media formulation is a key for your microbial growth as well as for your product formulation. Then when your media is sterilized, you have to provide this into seed fermenter as well as in production fermenters. And when your media sterilized and you are going to ready for fermentation at large scale, then you have to inoculate your media with your desired microorganism. Either your desired microorganism is fungi, bacteria, or yeast. What kind of microbe do you have, and what kind of fermentation you are going to do, and what kind of product you are going to produce. This all depends on your targets, what targets you are going to achieve. After this whole fermentation process, then you have to do further harvesting process. Here, before in my slide, when we are talking about fermentation classification, so and in fermentation classification, we have classified based on the mode of cultivation. So what kind of fermentation you are going to do? Either you are going to do batch fermentation, or either you are going to do continuous fermentation, or either you are going to do fed batch fermentation. This all depends on the type of product, type of microorganism, and product demand, industrial demand. If your product demand is very high, you can produce these in large scale and in continuous mode. So in this continuous mode, you have to recover your product with the passage of time. When your fermentation process is complete, you have to harvest the process. And in this process, the first step in harvesting in downstream processing is separation of cell biomass and metabolites. So here again, in fermentation process, there are two main steps. The first step is scale up, and or we can call it upstream processing. The second step is called scale down. You can call it downstream processing. And when we are talking about downstream processing, the first step is separation. And how we can do separation of microbial cells from liquid media? You can do this separation either by using centrifugation, either using your filtration process or something else. This all depends on what kind of facility do you have in your fermentation in your fermentation setup. After filtration, you can separate cell biomass, you can separate cell-free supernatant. If your target is to produce cell biomass, then you can move on further with biomass. And if your main target is metabolites, then you no need to do work with biomass. You can do work with your cell-free supernatant. And after cell-free supernatant, then you can further purify by different process, different techniques. And one thing here which you need to mention is deciding based on your target product. You first have to study either your product is intracellular or your product is extracellular. So we are going to produce biomass in case of intracellular products, or sometime if you need biomass for other process, other things, then you can work with biomass. Because in some cases, the first example is yeast biomass. As you all know that we are using yeast biomass in our daily life. In all kind of bakery item products, we are using fermentation through microbial biomass. But in some cases, biomass, this bacterial biomass or microbial biomass could be utilized as a antibiotic, as a medicine. So this all depends on what kind of activity you are going to do with your fermentation process. So this is the whole summary, whole process of fermentation. Either you are going to operate these process at lab scale, at pilot scale, or industrial scale. You must have to follow all these steps. So these are the main steps of fermentation process. So in the last step, last slide, we are going to mention the first one is stock culture. How we can get the source of microbe, where to get these microbes, because in all kind of fermentation, microbes is the key. When we are working with fermentation, you cannot do fermentation without microorganism. And for this micro fermentation, your main source is microbes. And how we can get this microbe? We can get microbe from two sources. Either you have to isolate from the environment, or you have to take this from culture collection. And if we are going to take microorganism from the natural habitat, so for isolation purpose, the microbes could be taken from three techniques. The first technique is shotgun approach. The second one is objective approach. And third one is enrichment approach. So these are the three different approaches by which we can take our microorganism. If we are talking about shotgun approach means you have to take random sampling from the environment. You are not going to specify. If you are taking your sample just randomly from the environment, that means this is a shotgun approach. And if you are going to specify your target, that means objective approach. For example, if you are going to isolate a bacteria or microbe which can have capability to produce cellulase enzyme, which can have capability to produce protease enzyme. In case of cellulase enzyme, how you can sample from which site? For case of cellulase, we can take sample from the soil having degraded wood material, having paper material, having wood logs. You can take sample from wood logs. You can take sample from the soil in which the paper is being degraded. You can take sample from the soil having fecal matter of animals. Why fecal matter of animals? Do you know? Because the animals feed on plants, and every plant, they are composed of lignocellulose. Their major component is cellulose. And you all know that cellulose is the most abundant polymer on the earth. So that's why we are going to take sample from the wood logs. If you are going to isolate cellulase producing microorganism, and if you are going to isolate protease producing microorganism, then you have to take sample from the soil where protein is being degraded in that soil. So guess what are the main sampling site for protease producing microorganism? What are the main different sampling sites? The first sampling site is where all meat industry waste is being degraded. Chicken waste, animal slaughterhouses. You can take sample from animal slaughterhouses. You can take samples from the dead animals' remains, the soils of the dead animal remains. And you can also take the sample from the soil where our all kind of hairs which are being degraded. And you can also take the sample from when animals becoming dry, the animal fell down in the earth, in the soil. You can take sample from that place. That means the soil in that place, they have microbes which have capability to degrade protein. So that's why we are taking the third one. The third approach is enrichment approach. In case of enrichment, you have to adapt your microorganism in the lab scale, in the laboratory. So here, here's is an example. You can see this is a soil sample. You make serial dilution, then make spreading on the plates, and after spreading, you can pour your sample, incubate sample in incubator, and after incubation, you can get different microorganism on the plate. And after that, you can further test your microorganism. Either your microorganism is meeting your objectives or not. Then you can have, you can test your microorganism on plate screening method. For example, in case of protease or in case of cellulase, you have to prepare petri dishes or petri plates supplemented with protein or supplemented with cellulose. If your media supplemented with cellulose or protein, and after incubation, the microbes being grown on that kind of media means that your microorganism have capability to produce zelanase, to produce lacase, to produce cellulase, to produce amylase, to produce protease. What kind of source, what kind of microorganism you are going to produce is all depends on the source of the media, on the source of the microorganism. So this all depends on the type of product. What are your main targets and what you are going to do. So this all depends on the soil. And if you are not going to do all kind of work, you think you have no time, you think this work is laborious work, then the other way is just get micro from culture collection. So every country have its own culture collection, and I think Indonesia, they have its own culture collection. You just can take your target microbe from Indonesia culture collection. Just take microbes, propagate in the lab, and do your fermentation. So this is the source of microorganism, and microorganism, they are very important for fermentation process. Without microorganism, fermentation process could not be accomplished. So this is all about source of fermentation, source of a microorganism. So now move towards next about microbial enzyme production. Why we are going to produce enzyme from microbes. What are the microbial enzymes? Because microbial enzymes are known to play a crucial role as metabolic catalysts, leading to their use in various industries and applications. And the end-use market for industrial enzyme is extremely widespread with numerous industrial commercial applications. So do you think now our 500 industrial products, they are being made using enzyme? So the industrial demand of enzyme is becoming high and high with the passage of time. So it's a time to boost your biotechnology, to boost your science for the mankind. So first here, this is in the first all slides, this is all about basic things. You must, if you are a microbiology student, microbiology researcher, or fermentation scientist, you must know some basics of all these things. So as an expert in this, my field of fermentation technology, so in my country, in my lab, I am working with some enzymes. The first I'm working with the cellulases. So what are the cellulases enzyme? So as you all know that cellulase enzyme, they can degrade cellulose. In the next slide, I will show you what is the cellulose and where it can be found. And cellulose is a polysaccharide, is a polymer. And why we call it polymer? Because they are made from many units of monomers. And the main monomeric unit of cellulose is glucose. And cellulose is a complex structure. for complete degradation of cellulose. So cellulases are the enzyme, and cellulases enzyme, they are categorized into three main groups. The first one is endoglucanase, the second one is exoglucanase, and third one is beta glucosidase. So all these three enzymes, they can work together to degrade a polymeric structure of cellulose into glucose. So endoglucanase means they can broken down the inside chains in the cellulose. If you have a look on the cellulose structure, they have some linkages, alpha 1,4 linkages, beta 1,4 linkages, alpha 1,6 linkages, beta 1,6 linkages. They have so many kind of linkages to combine the monomeric unit to form a polymeric structure. So these enzyme, exoglucanase enzyme, they are the enzyme which can degrade the cellulose chain from outer regions. And the last one is beta glucosidase. Both endoglucanase and exoglucanase, both enzyme, they act on cellulose to produce a shorter chain. As I said before, cellulose is a polysaccharide, and with the help of these enzymes, we are going to convert cellulose polymeric structure into monomeric structure of glucose. So the third enzyme is also a key enzyme, which is called beta glucosidase, and this enzyme is also known as cellulobiohydrolase enzyme. And this by the action of endoglucanase and exoglucanase, the cellulose polymeric structure is converted into smaller unit disaccharides in the form of disaccharide, in the form of oligosaccharides. And this beta glucosidase enzyme, this act on that small oligosaccharide units and convert this into a glucose unit, which is our ultimate target. So by combining all these three enzyme combined action, we can produce glucose from the cellulose. And as you all know that glucose is the main source of energy. All kind of microorganisms, they can produce all kind of living things. They can metabolize glucose. So we have to provide glucose as a main metabolic pathway. And if microorganism or any living organism, they can utilize glucose as an energy source to produce a variety of metabolites. And these metabolites, which we can further use for different kind of processes. So how we can produce these cellulase enzyme from microbes? A variety of microbes, they have capability to produce cellulase enzyme. But here,
Okay, Dr. Irfan, excuse me, maybe in 10 minutes more. Can you speed your presentation? I'm sorry.
okay. So I'm sorry, we have some time limitation. So to squeeze my presentation, I'm quickly gone through this all things. So this enzyme could be produced from bacteria like Bacillus, Pseudomonas, Clostridium species, and this can also be produced from fungi like Aspergillus species, Trichoderma, Penicillium. They all have capability for the production of enzyme. And in fungi, the most common is Aspergillus. Why we are more focused on Aspergillus? Because Aspergillus is more common in our homes, in our basements, in bathroom, in bedroom, in kitchen, in living room, in storage, in offices. They have a wide abundance and they are abundantly available in our environment. So these have a good opportunity to can produce cellulase enzyme. This is all about the structure of cellulose. You can see so how cellulose is made. Cellulose is many chains made up from glucose units. So all these enzymes, they work on cellulose to produce a glucose units. In the last my work, we have screened 6 bacterial strains. You can see different bacterial strains which have capability to produce cellulase enzyme. In this picture, you can see I have grown these bacteria on on CMC agar plates, and you can easily see that all bacteria, they have capability to degrade CMC in the media. That means they are capable of producing cellulase enzyme. So this is a qualitative assay which we have done using grams iodine, and we have identified Bacillus megaterium, Pseudomonas strepsleri, Bacillus aureus, Bacillus ligniformis, Paraligniformis, Bacillus flaccus, Bacillus verdimani. All these strains I have isolated from the Pakistan, and all these strains, they are further classified, identified using 16S genomic DNA and 16R sequencing technology, and all these sequences they are submitted in NCBI. You can check these session number from NCBI. You can find the data about my strains. So after these 6 strains, we have isolated from the soil, from the wood log soil, and have capability to produce enzyme. Then further, we have screened a variety of substrate, what kind of substrate they have capability to produce enzyme. So we have tried a variety of substrate like different substrate. I have used a wheat straw and I used a different bacterial strain, and you can see that 24-hour fermentation time is best for the production of cellulase enzyme. And then we have optimized the media through responsible methodology for the production of cellulase enzyme, and we have selected different variables for media components, and you can see that the maximum production of a cellulase enzyme CMCs is about 120 units IU per ml per minute, and for FPAs we got 125 units of filter paper activity per unit per time. And these graphs indicate that we have selected 6 variables as a media component, and we have to check which variable have significant impact on enzyme production. And we have seen that X4 and X1 and X5 and X6, they are the significant media component which can significantly affect the production of cellulase enzyme using Bacillus species. So these are some graphical representation. You can see that potassium dihydrogen phosphate and potassium dipotassium hydrogen phosphate, as well as ammonium sulfate and yeast extract. These are the key nutritional components which affects CMC's production. After maximum production optimization through RSM, we have characterized the enzyme, and you can see that the FPAs and CMCs, they are optimally active at 50 degree centigrade temperature, and both enzyme, they are active at pH 5, and we have checked the metal profile of all these enzymes, and you can see that mostly the metal ions, they are the activators, they are not inhibitor of this enzyme. And we also studied about the different effect of solvents, different chemicals, and all you can see there are different chemicals like SDS, 80, butanol, hexane, ethyl alcohol, isopropanol, methanol. All these chemicals they have a very less effect on our enzyme. That means that our enzyme produced in my lab, they are oxidants, they are surfactant stable, they are stable in different kind of solvents. And then we have studied different kinetics of the enzyme and thermodynamic properties, the Km and Vmax of the enzyme from Bacillus subtilis and Aspergillus niger, activation energy, delta S, enthalpy of the reaction, entropy of the reaction. We have measured all kind of thermodynamic property of the enzyme in my lab. And then we have applied this enzyme in 2 ways. First, we have applied our cellulase enzyme in terms of enzymatic hydrolysis of different biomasses. And second, we have used this enzyme for the biostoning of denim genes. You can see in this picture, you can clearly see the difference in texture, different in color of treated and untreated denim genes. So right now, the cellulase enzyme could be utilized in bioenergy application for enzymatic hydrolysis of different kind of biomasses. And this enzyme also be used as a in textile industry as a biostoning agent of denim jeans. So this is some work which I have published about cellulases in different journals for the period of the last 10 years. You can see different kind of publications from all about a cellulase enzyme using different substrate as well as different microorganisms from penicillium, from different sources. The second enzyme which I work on is proteases. As you all know that proteases is also a key enzyme which hydrolyzes the peptide bond and proteins present in important group of commercial enzyme applications. So microorganisms represent an excellent source of proteases because they are the main source of enzyme production. And although the proteolytic enzyme can be obtained from animals and plants, but we are more focusing on microorganism. And why we are more focused on protease enzyme? Because protease enzyme had a wide variety of application. It could be used in dairy industry, peptide synthesis, cell degumming, therapeutics, leather industry, waste management, pest management, contact eye lenses, and animal feed industry. A variety of industry are being used this enzyme as their industrial application. So that's why we are more focused on production of proteases for their industrial applications. And for this, we have screened different kind of media for the protease production like glucose, peptone, potassium hydroxide, MgSO4, NaCl, calcium chloride, and yeast extract. And among all these nutrient components, you can see that calcium chloride and glucose is very important for the production. And after this, we have then we optimize different concentration of these components, and we can get a different yield of enzyme production through optimization. And after this, we have characterized the protease produced through fermentation, semi-fermentation. We can see that our protease enzyme produced from bacillus is active at 55 degrees centigrade and active at pH 10. That means that the enzyme is thermostable and is alkaline in nature. And after this characterization, we have further purified the enzyme and studied their metallic profiles. And you can see that the enzyme is being modulated by calcium ions. So calcium ions play an important role in the stability of the enzyme. So we already almost know that most of the metal ions, they act as a cofactor for enzyme activity. So some metals, they act as an activator of the enzyme activity, but some chemicals, they act also as an inhibitor of the enzyme. So before we are working with enzyme, you must know your enzyme. What is the activity of your enzyme? Either your enzyme is activated by metal ions, or either it is deactivated by some sort of other metals. Then we have applied this protease enzyme in different application. The first application we have done as a detergent. And you can see that the cloth is being stained with blood. The first application, this protease enzyme could be utilized as a de-staining agent, as a stain remover. Every kind of stain or blood stain on your cloth could be washed through this enzyme. And the second application of this protease enzyme could be utilized in tanning industry, in leather industry, as a de-hairing agent. You can see that this is the skin of animals before and after treatment with the protease enzyme. You can see the hairs, they are being degraded by this protease enzyme. So this enzyme could have wide application in different industry like detergent industry, tanning industry, as a de-hairing agent, as a de-staining agent for different application. So this is all about protease enzyme, which I have worked in my lab in Pakistan. So this is some work which I have published in different journals about protease production. You can see different bacteria, different applications, purification and from different strains. So this is all about microbial enzyme. The third one, the last, I'm working about creatinase enzyme. As we all know that creatinases, it is also a type of protein type enzyme which can degrade keratin. How we can isolate in my one of my PhD student, she have done this work. She have isolated almost 7 bacterial strain which have capability to degrade protein. We have identified the strain and submitted in gene bank. In the last this slide, you can see that the selected bacterial strain have capability to completely degrade the feathers, and these feathers, they are all made up of keratin. Keratin is a type of protein, fibrous proteins. So keratinase enzyme, they're also very important for degradation of keratin. And as you all know that in our environment, we have so many kind of feathers being produced from animal feather birds. So these feathers could be, and almost we also have some hairs. You can see every territory, every person, they have to cut their hairs almost monthly. So this hairs, they are depleting our environment. They are contaminating our environment. So these hairs could be degraded by these creatinase enzyme to produce valuable things. For creatinase production, we have selected a different, we have optimized media components. You can see we have selected 10 different nutritional components for the maximum production. And we have found 4 parameters among these 10. They are significant for protease production. And for protease production, we have done optimization through response surface methodology as well as artificial neural network technology for optimization. And you can see these are the different plots of control plots indicating the maximum production of creatinase through responsible methodology. After maximum production, we have characterized these creatinase enzyme, and you can see that the enzyme is alkaline in nature and active at pH 9, and have maximum activity at temperature 60 degrees centigrade, because most of the industrial processes, they are being operated at very harsh environment. So we must make some sort of enzyme which are thermostable, which are alkali stable. And further, we have characterized, purified this enzyme, and we can see that our creatinase enzyme having molecular weight of about 60 kDa. And we have studied all kind of thermodynamic studies like temperature, dissociation constant, shelf life, deactivation value, enthalpy, Gibbs free energy, and enthalpy. And we have also measured about the kinetics of the enzyme like Km and Vmax. You can see that the Km of our creatinase produced by bacteria is having 9 milligram per ml and 61.58 micromole per ml per minute of maximum velocity. And then we have some bioinformatic analysis, and then we have some further characterization like metallic profile and some organic solvents, some detergents, and you can see that most of the chemicals, they have very less effect on our creatinase enzyme. That means our creatinase enzyme is activated by calcium ion. Most of the proteolytic enzyme, you can see that here, the calcium ion, they are very important. They act as a modulator of the enzyme. They act as activator of the enzyme. And all kind of alcohols, they have very less effect. That means our enzyme is organic solvent stable, and our enzyme also stable under different kind of surfactant agents like ASDS, EDTA, CTAB, beta-mercaptoethanol, tritone, X100, and 280. They have very less effect on enzyme. And then we have performed some bioinformatic analysis, some computation analysis, and you can see that we have found the catalytic active site, and we see what kind of amino acids they are present in the active site of our creatinase enzyme. And you can see that the most enzyme is histidine and the leucine, cysteine, arginine. They are most common, and they are abundantly available in active site. After this, we have performed some applications. We have performed application as a biofertilizer. So how we have done this? We in this picture you can see that we have used three kind of plant material in three different solution. The first one is this C is control. Control mean just soil and water. And the second one is soil irrigated with urea fertilizer. And the third one is FH solution, mean feather hydrolysate solution. So in this part, we have supplied feather hydrolysate to the plant, and in all three application, in all three pictures, you can see there an incredible length increase in the length of the plant is being observed, and this indicated that this material, this enzyme could act as a biofertilizer. So how we can use this? You just produce creatinase enzyme, just collect feather hydrolysate, or just collect the raw hairs from the environment, hydrolyze them with produce creatinase enzyme, and then this solution could be utilized as a biofertilizer to different kind of crops. So we have used three different plant material, and you can see that there's a significant difference in plant length as compared to control and feather hydrolysate urea. So this experiment indicated that the creatinase enzyme produced in my lab, we have capability to act as a biofertilizer to make the environment sustainable as compared to chemical fertilizers. So some of the data is not published yet. I have published a very few part of this creatinase enzyme, and you can see just I have published about creatinase isolation, identification, and screening and production optimization. Just one part of this is published. So so this is all about enzyme production at lab scale. How we can produce this enzyme at pilot scale? What are the main key issues? If you want to produce enzyme, if you want to do fermentation at large scale, at pilot scale, you must have to focus on different issues. There are some issues which are being taken and during operation and during designing. So if you are an engineer, if you are a chemical engineer or biochemical engineer, you must have to focus on these 4 points. The first one is good manufacturing practice regulation. If you are doing fermentation at large scale, industrial scale, you must make your process in a good regulation. You must be adhere to good manufacturing guidelines. And the second one is 3 Qs. What are these 3 Qs? These 3 Qs are, number one is design qualification. You must know about the person who is designing your fermenter. Is he have enough qualification to design your fermenter? Does that person know about fermentation? Does that person know about microbiology? The second one is installation qualification. When you have purchased a fermenter in your lab, who will install this fermenter? Either you have ability to install your fermentation, your fermenter, or you can hire a specialized person who have a well-defined qualification to install your fermenter. And the third one is operation qualification. Who will operate this fermenter? Does the person who will operate your fermenter is enough capable of doing all these things? What degree he has done? Is he an engineer? Is he a biotechnologist? Is he or she is a chemical engineer? Or is she have a knowledge about microbiology? So all these 3 Qs, they are very important to commercialize your production at industrial scale. The third one is validation. What kind of protocol you are using in your fermentation process? Is your protocol is an established document evidence? You must have to be careful about this. And the third one is, the fourth one, the main key issue in installing is about current employment. Employed, they are strictly in drug industry where stringent requirements are made. So you must have to follow good manufacturing practices. You must be careful about the handling of the fermenter. You must know about the safety precautions. Your employee must know all these matters before going to produce. And the second important the issue in pilot scale production is bioprocess parameters. So what are the bioprocess parameters? Shortly, the main bioprocess parameter which can affect your fermentation process is temperature, water availability, carbon source, pH, salts, oxygen, gas evolution, vitamins, product byproduct removal. So all these parameters together make your process very complex. So before going to make your process complex, you must know that in majority of fermentation process, an unusual foam is being produced. So you must have to deform your media. How to deform your media? You must use some antiforms. So what kind of antiform could be utilized for fermentation process? So what is an ideal antiform? What are the main properties of an ideal antiform? An ideal antiform must be readily and rapidly dispersed with rapid action and must be high active at low concentration. And your antiform must have prolonged action. It should be non-toxic to fermentation microorganism, humans or animal. And it should have low cost, it should be somewhat stable, and it should have compatibility with other media components and the processes. For example, having no effect on oxygen transfer rates or downstream processing operations, because if the foam is being produced in your fermentation media, it will ultimately adversely affect the oxygen ratio, and most of the fermentation process which are being operated at industrial scale, they are aerobic and sometimes they are anaerobic. So be careful about your fermentation process. So all these parameters we must need to focus about fermentation at pilot scale. So these are some fermenters. This is the first 1 liter fermenter I have worked on protease production using this fermenter, 1 liter. And this fermenter is present in my country in PCSAR lab. Before I have worked 4 years with these process. And this is the second fermenter about 10 liter capacity. And this one is a 30 liter capacity fermenters. And the last one is a 500 liter fermenter which are being installed in my country. So in short, this is all about the summary of this whole process parameters. We are more focusing on enzymes, and you all know that enzyme, they are multi-step processes, and you must be careful what is your source of product. You can take your enzyme from animals, from plants, from bacteria, from fungi, from yeast, and then you can produce the enzyme. After getting enzyme, you have to check the catalytic property of your enzyme, because every enzyme has its own pH, temperature, metallic profiles, other activities. So before using an enzyme in commercial application, we must know all about catalytic properties. And if your enzyme catalytic property is not very well, then you have to improve your enzymatic catalytic properties. So what kind of catalytic characterization we need to improve? We need to improve kinetics, we need to improve activity, we need to improve stability, shelf life, structure, all things. And for this, we have to make genetic engineering, we have to make engineering the enzyme, and we have to reevaluate our enzyme. And how we can reevaluate? You can enhance the catalytic efficiency, you can enhance the activity of the enzyme, you can enhance the stability of the enzyme, you can enhance the shelf life of the enzyme. Because the main problem in fermentation process in biocatalysis is that when you have produced your enzyme, after certain time period, the activity is going to be slowed down. It's becoming reduced activity. So you must think about that, how we can uh, make your enzyme more efficient, more stable after a long period of time. And your enzyme should be stable under extreme environment. So majority of the industrial processes which are being conducted in industry, they are working at harsh environment. They are working at high temperature. They are working in high pH, in extreme environment. So by the aid of protein engineering, by the aid of metagenomics, by aid of genetic engineering, we can alter our choice of gene and insert this gene to choice of microorganism, so that you can produce enzyme of your desired targets. And all enzyme who are working in industry, they must be stable, they must have a long shelf life, they must be active at high temperature, they must have a good kinetic parameter, so your enzyme could be effectively work in industries. So this is all about this is all my research group. This whole work which I have presented here is being presented, done by some my PhD student, some my master student, and I have some collaborator from Brazil, from other universities also. And I am also wishing and hoping, when I will present my work in any other station, here some picture will be from BRIN from Indonesia. So I hope for the best. This is all from my side. Thank you very much. Shukriya. Terima kasih. This is all from my side. And once again, I'm very thankful for the administration of the BRIN, all people from the BRIN who are assisting me, and the government of the BRIN and government of the Indonesia who is supporting all such kind of activities, such kind of program. Thank you very much. If you have any query, just raise your hand. Thank you very much.
Okay, thank you, Dr. Irfan. Very nice presentation. Okay, ladies and gentlemen. So we are go to the discussion session. Okay, there is some question in the chat. I will read for you, Dr. Irfan, from Mr. Norvianto. I want to ask to Dr. Muhammad Irfan, one, how is the application of omics studies in determining the best microbial strains that can produce enzyme very well? And number two, for pilot scale or mass production using bioreactor, what type of bioreactor is suitable for enzyme producing microbes with specific fermentation process, and the need for suitable microbial strain is in that bioreactor condition? Thank you. Okay, Dr. Irfan, you can answer.
Yeah, the first question is about how we can select our target enzyme, target microbes. So for the first question, if I have told you before, first select your target. What kind of enzyme you are going to produce? You think, let's suppose you are you want to do work on amylase enzyme. Then you must have to isolate, you have to take sample from the sites where starch is being degraded. And how we can know that which place starch is available? You can easily see from the soil where potatoes they are dumped, potato waste being dumped in the soil. And you can also see the places where the maize, corn starch is being dumped, and also from maize industry waste is being dumped in which places. You can take sample from that place, and then spread your sample or load your sample on a plates supplemented with starch. And when you have spreaded your sample on that plates, maybe if you can get microbial growth on that place, that means the microbes have capability to utilize starch as a carbon source. That means the microbes have capability to degrade amylase enzyme, and this amylase enzyme have capability to degrade starch to produce some products, and then the microbe can grow on that place. After this, further you have to confirm it whether your microbe have capability to produce amylase or not. Then you can do some qualitative test. What are the qualitative tests? Qualitative test means you have to screen your microbes on a uh, medial growth, then stain with some staining agent. For example, in case of amylase, you can stain with iodine. For in case of proteins, in case of protease, you can spread your microbe on protein, on casein or skim milk plates, and then spread with stain with TCA. And in case of cellulase enzyme, you can do staining with uh, iodine as well as Congo red. And after staining, you can see in all these methods, you can see a zone of clearance, a halo zone around microbial colonies. That means the microbes have capability to produce uh, enzyme of your target. Did you got my point? I think the first question answer.
Okay, Mr. Nurfianto, maybe you want to ask to the Dr. Irfan, or it is enough? Begin to the next question, number 2, for pilot scale or mass production. Dr. Irfan, please.
For pilot scale production, I'm using still tank reactor for industrial fermentation. But if your target microbe is fungi, then you cannot use a stir tank reactor. For fungi, you have to use a airlift fermenter for the production of enzyme at industrial level. Because the fungi, they are filamentous, and when these filaments, they they strike with the impellers of the blades of the fermented tank reactor, the filamentous fungi, they will cut down into small pieces. And then the filament microbes, the fungi, they cannot grow well in the media. So you have very less production. So mostly in case of bacterial fermentation, we are using a stirred tank reactor, stirred fermentation system at pilot scale, at large scale. But I have not used these stirred tank reactor at large scale, like 100 liters or 500 liters. I have not used this before, but I have used about 5-liter fermenter or 10-liter fermenter at large level.
Okay, for because the time is limited, I go to the next question. Is from Dian. How can we avoid batch variation? How can we avoid batch variation of biological or biosimilar production of microbes for any acceptable range for batch variation? Okay, Dr. Irfan.
Sorry, can you repeat again?
How can we avoid batch variation of biology or biosimilar production of microbes or any acceptable range for batch variation?
Yeah, for batch production, you must have to check about your microbes and the type of method you are using. Because as you all know that every microorganism have 4 different growth phases, either using on lag, log, stationary, or decline, or decline stage. So most of the secondary metabolites, they are being produced at a stationary phase. So we have to determine what is the growth phase of that microorganism and how long this growth phase we lost of a typical microorganism. So in this way, we can select either we have to continue in batch, or we have to continue in a fed batch, or we have to continue the whole fermentation process. Maybe I think
Okay, is enough. Or maybe Dian want to raise hand, maybe, or is enough? Okay. From Ima, could you please elaborate on the challenge that Pakistan faces in the scaling up microbial enzyme production through fermentation? In case in Pakistan, I'm sorry.
We have some issues because as a nation, we have our different challenges as compared to Indonesia. So it's some difficult for us to produce these enzymes at a large scale.
Okay, there is someone raise hand. Please speak, Mr. Edi Wahyono.
Okay. Thank you, Dr. Muhammad Irfan, for your nice and comprehensive presentation on fermentation technology. I have 3 questions for you. First is, when we use certain media, and we have fermentation which a little bit turbid in our broth, and it would be difficult to measure the growth of the biomass by using a spectrophotometer. So is there any approach, as any method as an approach to measure this growth of the media using spectrophotometer, because sometimes we have difficulties to solve this situation, to face this situation. And the second question is there any industrial enzyme, commercial enzyme industry in Pakistan, so far, so to fulfill your industrial needs in Pakistan? And the last one is, have you experience to deliver your research results to industry for commercialization of your enzyme resulted from the your research results so far? Thank you.
Okay, thank you very much for your nice questions. The first question is in case if you are going to measure cell biomass during fermentation process. So please keep in mind if you are working with the bacteria or fungi and you are very much interested in biomass estimation and biomass working, then your media component should have no insoluble component in your media, right? If you are not going to measure cell biomass, you are just focusing on the metabolites, then your fermentation media might have some insoluble materials like insoluble nutritional components like agricultural waste, something else. But if, as you said, your media become more turbid, and it is very difficult to measure by spectrophotometer. The second option, you can go towards measuring cell mass using filtration method, or you can measure dry weight method. Just take sample from fermentation system. You can centrifuge that sample and then measure the cell biomass, dry weight basis. Or you can pre-weight filter paper, then put your sample on that filter paper, the cell biomass will be on the top of filter paper, and then you can measure it again. So by this way, you can measure on dry weight basis. This is the only option. The third option is that if you are going to measure this, then you have to check the cell wall component. For example, in case of fungi, in case of solid state fermentation, in solid state fermentation, your media should be in solid. And if you are going to measure the microbial growth, then you have to check glucosamine content in the cell wall. How much glucosamine content, how much cell wall component is being present at that time. So in this way, you can measure the growth of the microorganism. You can make growth curve by measuring cellular component. Did you got my point? I think.
Yeah, the problem is, let me say, if we measure like that, with it would be there is yeah, actually we ever had the fermentation using corn cob media.
Yeah.
So that's a big problem, even though we do it centrifugation, something like that. So sometimes the cell will mix up with the corn cob.
Yeah, but in that case, it is
Yeah.
impossible to measure. It is impossible to measure the
Okay.
cell biomass.
Okay.
Yeah.
Yeah.
Second question is there any country, is there any commercial enzyme industry in Pakistan? Unfortunately, right till now, we don't have any commercial enzyme industry in Pakistan. You see, we are very neighbor to China, and China is a good friend of Pakistan. So most of that chemical, most of these things, they are being shipped from China to Pakistan. And we are also working with closely with China to develop some industries in Pakistan. Maybe you have heard about CPEC project. So mostly industries, they are being established in Pakistan with China and Pakistan cooperation corridor through these projects. And the third question answer is that right now, so far, I did not contacted my research in industry, but at very small scale, in another research center, we have applied these experiments.
Yeah, I mean, from your research,
Yeah.
so you haven't been you haven't delivered to industry for commercial production, something like that.
Yeah.
You haven't done that yet.
No, because if but this question answer is not relevant. Maybe something is relevant. I'm still have a long life with the mercy of God to work more. You see, I'm just too young now, 38 years
Okay.
old. I have a very
Okay.
productive time ahead. Maybe I will transform this research to industry.
Yeah.
And
I just want to know whether
no,
you
no,
have
no,
an
no.
experience on that
Not experienced
issue.
yet. Not
Okay.
experienced yet. Because we do have some problems in my country, like political instability. So some kind of that issues have. So due to this, we are not able to transform these things.
Okay.
Yeah.
Thank you, Dr. Muhammad Irfan, for your answer. Thank
Yeah.
you.
Thank you very much. You are welcome.
Okay, thank you. Because we limit time, I will read the conclusion. Okay.
No,
For
there
one,
are some questions.
okay. For one, fermentation is the success key for enzyme production, and for pilot scale, use cheap material to meet economical value. And design fermentation depend on what your target. Okay, thank you. I will giving time to Mbak. Okay, please. Thank you.
Okay, thank you, Dr. Titin, for leading a very interactive discussion. And thank you, Dr. Muhammad Irfan, for a very interesting presentation. And as
Thank
a token of our appreciation, we will give a certificate to Dr. Muhammad Irfan as a speaker. Thank you. Thank you very much for sharing your valuable knowledge. Okay,
Thank you.
ladies and gentlemen, we have finally come to the end of part of this webinar. Before I close this session, I would like to ask Head of Research Center for Applied Microbiology, BRIN, to give us some short closing remarks to Dr. Ahmad Fathoni. Time is yours. Okay, Pak Fathoni. Are you still joining with us?
Yeah.
Okay.
Okay, sorry, I joined mobile. Sorry, can you see?
Yeah, Pak.
Hello, yeah. Okay, thank you very much, Dr. Muhammad Irfan, for your sharing work and expertise on enzyme and fermentation technology. And again, everyone, Bapak Ibu, again, I emphasize that we need these two, yeah. We need these two expertise more in Indonesia, enzyme and fermentation. So please, maybe if here there are our partners, yeah, from university, especially students, yeah. So please, next year we will have a project focused on bioprocess engineering and then doing like a enzyme production, and also we are now developing new laboratory in Cibinong for a fermentation process. So we invite you all to join us for even for research or for a studentship, yeah, for student. Yeah, we BRIN also provide a master and PhD scholarship through degree by research program. Yeah, again, thank you very much for everything, ya, Pak Irfan and Bapak Ibu sekalian. I hope that this webinar today is useful and uh, a good start for us to collaborate more in the future with Dr. Muhammad Irfan, yeah, our team in Pakistan and here in BRIN. Yeah, this is again first time for Dr. Irfan
Yeah.
visit BRIN, visit Indonesia, and I hope to see you next year or in the next occasion. We can also hopefully visit your team in Pakistan, and also you may visit again with your team to BRIN here in Cibinong to collaborate more in enzyme research and also enzyme production. Yeah, thank you, everyone. So I close this webinar today and see you in the next webinar. Assalamualaikum warahmatullahi wabarakatuh. Thank you. Okay,
Waalaikumsalam.
have a good day, everyone. Bye-bye.
Thank you very much.
Thank you, Pak Fathoni. Ladies and gentlemen,
Bye.
this is the end of our agenda for today. Lucy Arini, on behalf of the committee, I would like to say thank you very much to our speaker, moderator, and all participants. And for your information, you can check out our latest information about PRMT at our social media on YouTube at PR Microbiologi Terapan BRIN channel and Instagram at PRMT_page. See you in the next event. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Yeah. Thank you, everyone. Okay.
Terima kasih.
Makasih Bapak Ibu tim semuanya ya, tim WiFi dan moderator MC.
Ya, terima kasih izin live.